Okay, some of the examples of the transgenic plants: BT cotton, BT corn. BT cotton is the contain the genes from the Bacillus thuringiensis, making them resistant to the insect pests. Golden rice, genetically modified, produce the beta carotene, a precursor of the vitamin A. Roundup, the ready soya bean engineered to the tolerate glycophosphate herbicide. What are the benefits of the transgenic plants? Increase in the crop yielding, resistant to pest disease and the herbicides, reduced need for the chemical pesticides, enhance the nutrition value. What are the concerns of the uh, pa- transgenic plants? These are the potential environmental risk, loss of the biodiversity, possible effects on the human health, ethical and the regularity issues. What are the pathogenic plants? Pathogenic plants are the causes disease in the other plants, animals, even the humans. Or the term is commonly used in the plant pathogens, microorganisms such as the bacteria, fungi, viruses, and nematodes that infect the plants causes diseases. Types of the plant pathogens: the fungal pathogens, Phytophthora infestinces, the Fucsinia graminosum, and next one is the bacterial pathogens, Xanthomonas viridae, and next one is the agrobacterial influences cause the crown gall diseases, bacterial pathogens, viral pathogens, tobacco mosaic viruses, potato viruses, nematodes, mineral species, effects of the pathogen plants. What are the effects? Reduce the crop yield and quality, economic loss of the agriculture, can spread rapidly under the favorable conditions. What are the control methods of the pathogen plants? Is the disease resistance plants varieties, crop rotation and the proper field sanitation, biological control using the beneficial microbes, chemical control fungicides and bactericides. Would you like more details? What are the mainly transient plants? The many benefits like the improved and the more efficient the weed control and decreased loss due to the insect pests, the viruses. The main hindrance of the higher yield of the sustainability, accessibility, the plant growth. This is caused by the fungi, bacteria, and the virus. Some of the insects, nematodes also. Main is the crop loss may be up to 30% of the due to the attack of the pathogens. Moreover, the development of the disease-resistant varieties and the fungicides, bactericides is reduced the considerably this happiness due to the changed genetic set of the disease-resistant plants. Plant viruses cause the serious pests in the microbial pathogens. Genetic information can be boost plant breeding efforts for developing disease resistant varieties. The genetic manipulation carried out of the plants for the production of transgenic plants have been described. What are the resistant to the fungal diseases? Some of the resistance to fungal disease. Several agricultural crops are the several damaged the fungal infections with the results of the dramatic yield loss. The spread of the fungal disease can be combated by the conventional strategies like the breeding for the resistance by the variation of the cultural practice and the fungicide treatment. However, the promising alternation that has the emerged recently is based on the strengthening of the endogenous, the deficiency of the penetration. Mainly, some of the systematic acquired resistance are to fungal challenge. Plant molecular biology is the biotechnology techniques taken the rapid progress, the identification and the cloning genes involved in plant deficiency response. Transgenic plants have the produced through the different strategies. What is the enhancement of the plant's structural defense, neutralization, the fungal toxin exploration of the antifungal genes from the non-plant sources. Significant reductions of the fungal disease in many cases have been observed by the Some pathogenic related proteins, PR proteins are the low molecular weight proteins, where the accumulating the significant levels in infected plant tissues. The major classes of the plant pathogenic proteins are the PR1, PR2. PR2 is also called as the B113 glucanosis. PR3 is the chitinase. PR4 is the heaven-like. PR5 is the thymine like the osmotin. The ability of the hydrolytic enzyme to break chitin and the glucan in the cell walls of fungal pathogen has been exploited to develop resistance against the fungal pathogens. Chitinase, plant chitinase accumulation can provide the protection against the fungal infection. Regulation of the chitinase genes takes the place during the development stage, but stress conditions such as the fungal attack can trigger the expression. When the cause of the fungal inoculation enhanced the activity was the noticed in the deficiency response the targeted towards chitin major cell wall component in the fungi. B1 are 3 glucanose. The in vitro experiments show there the alone or combination of the chitinase and B13 glucanose inhibited the most fungi tested. The P pathogen, the Pfizeria solanae, strongly inhibited by the chitinase, and B1 is the 3 glucanose, indicating that these enzymes can be employed as in the weapon of the combat several pathogenic fungi. High activity of the chitinase and the B13 glucanose is the frequently found in the native plants, but substrate for the enzyme is absent in the plant. Similarly, substrate for B1 glucanase is callose is the usually present only in the small quantities. Availability of the substrate in abundance of the could be present only in cell wall many fungi. Since the chitinase is the induced upon the infection with the different kinds of pathogen, it has been the proposed that the participated in the active defense response of plants. At least 5 classes of the plant chitinase as well the characterize the base of the sequencing alignments or amino acid sequences. Class 1 chitinase one contains the N-terminal crystal, crystal rich amino acid. Transgenic techniques that provide the excellent opportunities for the plants on the combo to the fungal attack. High level expression, the chitinase and 1,3-B glucanose transgenic plants result in enhanced levels of the fungal resistance. Expression of the class 1 catalase gene, the activity of the endogenous homologous genes in tobacco plants results the disease resistance the transgenic plants. Activation of the phyto, phytoalexins. Phytoalexins are the low molecular weight antimicrobial activity secondary metabolites produced in plant response infection with the pathogen. Reversal and one of the commonest stimulation the phytoalexin is synthesized by the key enzymes. Stibulin synthesize transformation of the rice with the stibulin synthesis gene STS of the VST1 gene from the grape has improved the resistance to the Prickleria oryzae. Similarly, STS sibling synthesis gene is the was transferred to the rice, brassica, peanut, tobacco. No more recently, the role of the mitogen activated the protein kinase cascade, especially in Nikon in the regulation of the phytotoxin synthesis, the responsible for the blast infection, manipulation of disease resistant genes, introduction of the resistance genes from the plant variety resistance the certain pathogens into the subscription varieties is one of the strategy. Late blight of the potato caused by the Phytophthora infestances is one of the most devastating fungal diseases in potatoes. Several origins have been identified and isolated from the various sources. The lipone gene has been isolated from the various sources of the stimulate innate immunity of Solanum species. Some of the hypersensitive response caused by the lipo, the source of the origin is the RPI BLBL was identified. The the transist expression of the lipoproteins, the RPI BL resistant gene nicotinia benthonema. The next one is the lead rapid identification of the RPI stole RPI PTL genes. The similarly is the RP, CF2, CF3 from tomato, engine from the tobacco, and L5 and XA21 is from the rice has been cloned, resistant the bacterial diseases. Okay, conventional breeding of effects have not the resulted the generation where varieties the resistance the bacterial disease due to the lack of the known resistance. One approach of the control bacterial disease is to improve the plant's defense against the particular pathogens like antimicrobial peptides. Antimicrobial peptides have the broad range of the antibacterial properties and then play the important role of the plant defense. Antimicrobial peptides with the helical structures are the ubiquitous found in many organisms. AMPs are the selective for the prokaryote membranes over eukaryotes membrane due to the predominantly negatively charged phospholipids in the outer left let of the prokaryote. Lysozymes. Genetic engineering offers some good protection of the against the bacterial attack. In one of the classic case studies of the novel gene encoding the T4 lysozyme was successfully expressed in many model plants. Model plants. Overexpression of the T4 lysosome can be combat the bacteria during the infection. T4 lysosome is the very effective against the wide range of the gram positive and gram negative bacteria with the specifically degrade the peptidoglycan, the bacterial cell wall. The lysosome genes have been the used for the confer resistant the transgenic potato against the Echerichia cartovara, which is cause the bacterial soft rot disease, transgenic apple plants with the T4 bacteriophage. Uh, Next one is the ceocropin. Ceocropin is one type of the is the strongly mainly the broad spectrum bacterial pathogenic expression by the cytogenic antimicrobial properties. For the past 2 decades, the persistent effort have been the made in the discovery of large group of natural antimicrobial peptides. Caps are generally two classes in the helical peptides, such as the serocropins and the magnonins, and second B sheet peptides, such as the defense, the protogenes, and the tetranes. These natural peptides exhibit the greater degree or broader biological activity, killing the bacteria, fungi, and extending the protozoa and concentration from the zero point 0.2 to 4 micrograms per ml. Cercopins the proceed the, from the giant silk mouth of the Helopathoceropora. The peptides interact with the other phospholipids membrane, the both gram negative and the gram positive bacteria, modified them from the large number of transit ion channels. Transgenic tobacco plants expressing the syringe have the increase the resistance of the peptidosyringe sarcotoxin. Sarcotoxin is the 1A is the antimicrobial peptide. The sarcophagopersogen is the self-defense system in the insects, protects the wounded larvae from the bacterial attack. The generate resistance plants the pathogenic bacteria. Sarcotoxin 1A gene was introduced in the tobacco driven the PR1 promoter activated by the salicylic acid pathogen infection, provide the protection against the Pseudomonas syringae, Irvinia cerevitova, and Xanthomonas campestris. Engineering toxin intensive genes. The toxin inactivating enzymes have been successfully used to develop transgenic plants. The bacterial hollow blight pathogen being Pseudomonas fasciculata produced the tripeptide toxin. Physotox inhibits the enzyme resistant to the insects. Bt gene and the toxin Bacillus thuringiensis is many species of the bacteria produce the protein in abundance when insect larvae ingest the bacteria with with their food protein present in the bacteria kills the larvae. The most of the widely studied is the bacteria is the Bacillus thuringiensis. This species also lives all over the wall when the bacteria form spores also from the large crystals like structure of the bacterial cytoplasm, which is made in the out of the protein. BT plants, transgenic plants are produced by the transfer of the gene from the Bacillus thuringiensis, coding for the crystalline protein. These cryoproteins are toxic to insects but non-toxic to human and the animals. Bt genes encoding the insectidal cryoproteins have been the transferred to relevant crops to confer the protection against their most important insect pests. Cryoproteins, once ingested by the insect, are the solubilized in the mid gut and then the claved there thereby the digestive proteases. Some of the resulting the polypeptides are the able to hint to mid gut of the epithelial cells, the reporters resulting cells was lysis and the finally insect death. Currently, but this is the major transgenic plants. For example, lectins, these are the carbohydrate binding protein found in many plants, the tissues, and the abundant in the seeds and the storage of the tissues. Some of the plant species, plant lectins are the particularly effective against sap-sucking Hemiptera. The toxic effects of the different lectins range from the south delay in the development of the high mortality in insects larvae. Therefore, enhancing their presence in some of the plant tissues may have insect tolerant effect. Transgenic rice with the Galanthus nivalis snow and agglutation gene has shown the resistant to brown plant hopper. And similarly, helium leaf agglutation possesses the anti-insectile activity different plants. The helium leaf agglutination gene was the transfer to the rice and the transgenic plants showed the resistance to the hopper. Protease inhibitors, PI, plants that contain the peptides acting the protease inactivating proteins. Plant protease inhibitors are the able to protect plants against insect attacks by the interfering with the proteolytic activity in insects. Digestive gut among their protein PS1, the serine and the cysteine PIs are the abundant in plant seeds and the storage tissues and may contribute to the natural defense system against the insect predation. The first PI genes that was the successfully transferred to plant species, resulting in the enhanced insect resistance, was the isolated from the copy and the encoded in the trypsin inhibitor, and transferred to the tobacco and protects these the plant species. Several insect species are the commercially released in the China 2000 in occurring the approximately 15% of the grown cotton is in 2005. Whereas the cystine 1 is the well studied in the cystine P1 from the rice seeds was with the being successfully introduced into the several different crops like the rice, wheat, oilseed, and alpha amylase inhibitors. Alpha amylase inhibitors are the attractive candidates for the control of insect pests. Alpha amylase. Next one are hydrolytic enzymes that catalyze the hydrolysis of the one-fluor glycosidic bonds to polypeptide hydrides. There are the present in the microorganisms, animals, and the plants. Alpha amylase are the most important digestive enzymes of many insects, which feed the exclusively or on starch products. Inhibition of the alpha amylase impairs the digestion in the organisms and causes of the shortage of the free sugar alpha amylase inhibitors are the found many plants in the part of the defense system in the abundant cells in the legumes. Resistance to the viral diseases. Antisense RNA approach. The viruses are the potential against the affecting yield many crop plants. The conventional methods such as the breeding for the resistance, heat treatment, or the some enforced the viral pathogens. Numbers of the strategies with using the molecular technology to develop integrate new resistance the factors against to plants viruses. Antisense RNA approach. The antisense approach attempts to block the replication of the viruses by hybridization of complementary sequences to the replicase viral gene. The first 250 nucleotides of the replicase gene of the Gemini viruses, wheat dwarf, mosaic viruses, completely inhibited viruses replication similarly. Tobacco gene was the integrated with the complete. Okay, next is the viral diseases, mainly virus resistance, the viral diseases, antisense RNA approach. The antisense approach is the attempts to block the replication of the viruses by hybridization of complementary sequence to the replicate the viral gene. The first 250 nucleotides that replicates the genes of the Gemini virus, weed or mosaic viruses, and next one is the tomato golden mosaic viruses. Then several lines that were reported in the exhibit the higher levels of the resistance, ribosome inactivating proteins, RIP. Next one is the virus code proteins. This is a mediated the cross protection. In this, the concept of the protection of the ability on viruses, the prevent inhibit of the another virus. Transgenic tobacco expressing the tobacco mosaic viruses, code protein showed the genes from the different viruses groups have been expressed with the transgenic plants. Code proteins initially point of the infection. The resistance showed the reduced number of infections sites in inoculated leaves, suggesting that the initial step of the virus's life cycle is what it has been there. demonstrate in the TMV across the production. Okay, these are the pathogenic plants. The next topic is the improved growth, especially genetic manipulation is the improving the natural quality of the plant products and the great importance of the plant biotechnology. Some successive has been the achieved the direction through the conventional crossbreeding plants. The advances made in the plant genetic engineering improvement of the crop yield and the quality has become the reality. The crop yield is the primarily depend on the photosynthetic efficiency and the harvest index. The quality of the crop depend on the wide range desirable characters like the nutritional composition of the edible pods, flavor, processing quality, shelf shelf life. These are the examples of genetic engineering improved growth and the golden rice. This is a provitamin A enriched the rice. This is one third of the world population is the depend on the rice staple food. The milled the rice is the usually consumed almost the deficient of the beta carotene, provitamin A. As such, vitamin A deficiency major nutritional disorder the worldwide, particularly in the people subsisting on rice. To overcome the vitamin A deficiency, it was the proposed in the genetically genetically manipulate the rice produce the beta carotene in the rice endosperm. The presence of the beta carotene in the rice gives the characteristic yellow orange color and the provitamin A enriched rice the approximately considered the golden rice. The genetic manipulation to produce the golden rice required in the introduction to the three genes encoding enzymes. The phytotin synthase carotene destroys the lycopene. Golden Rise is has met the almost the all opposition the rise of the opponents of the GM foods, or the people are the still against the large scale production of the golden rice, as this will open door the entry into the many other GM foods. In 2004, the group British scientists have developed the improved version of the golden rice. The new strains of the golden rice 2 contains the more than 20 times of the amount of the provitamin A, that is the precursor. Disclaimed the daily consumption of the 70 grams rice to meet the recommended the dietary allowance of the vitamin A. Amino acids of the seed storage proteins. The genetic engineering the extend the shelf life of the fruits. Mainly is the development of the flower tomato. The genetic manipulation of the fruit ripening become important commercial aspects. The plant genetic engineering ripening the fruit activities process the characterize the process of the fruit gets the softened results the activity, the cell wall degrading enzymes, namely polygalactose and the pectin methyl enters the phytohormone. The ethylene production is the intimately linked to the fruit ripening, the triggers ripening process. The fruit addition of the exogenous ethylene, the promoters of fruit ripening, while inhibits the ethylene biosynthesis and drastically reduce the ripening. Development of the flower sour tomato, manipulation of the enzyme polygalactose, and explain the mechanism of the PG antisense RNA approach, longer shelf life of the fruits and vegetables. Mainly are the spoilage of the fruits and the vegetables, the sense the pickled flowers, collectively referred as the post-harvest spoilage, the major concern in the agriculture. This hampers the distribution of the system is particularly when the transport is done to the far off places. The successfully manipulation of the delay ripening sensation and the spoilage of the various foods will significantly contribute to the appropriate food distribution of the good economic practice in agriculture. Suppressing of the biosynthesis of the ethylene appears to the promising area as the reduce the spoilage of the fruits, vegetables, and senses of the flowers. The three different strategies, the black ethylene in the senses of the tomato have been described. The same approach is successfully used for the fruits, vegetables, and etc. Manipulation of the ethylene biosynthesis, antisense gene of the ACC oxidase, antisense gene of the ACC synthase, insertion of the ACC demonized genes, fruits and the vegetable decolorization of prevention, mainly genetic engineering for the improvement of the flower pigmentation, genetic engineering for the male sterility, mainly all are the aspect of the improved growth. Next is the edible vaccines. The edible vaccines are the transgenic plants in the animal based to production, or these are the contain the agents of the trigger animals immune response of the. In simple terms, the edible vaccines, the plant or animal made the pharmaceuticals. The concept of the edible vaccines is created by the Charles Artner, Texas University, when she saw a mother offering up the piece of the banana as the crying baby. The advantage of introducing genes the plants then the motor the idea of food could be genetically altered, produce the vaccine their edible parts. Vaccines have been the developed for over 70 infectious diseases that the strike people at the every stage the life. A recent advance the way vaccinology as the created and the array of the novel vaccines lived in the non-living antigen and the delivery systems. These are the administered the basically via painful injections process. When the mass immunization was undertaken the developing countries, it was the imperative and the produce bulk vaccine. Methods of the developing of the inedible vaccines. Mainly the selected gene obtained from the microbes encoding the specific antigen can be handled two different ways. One is the suitable plant viruses genetically engineered produce the desired proteins. The recombinant viruses then the incorporated into the plant, which is the enables the produce the huge number of new plants, which is the chimeric version variants in the isolated in the purified. The consequential edible plant vaccines can be the used for the immunological applications. In another method of the desirable genes is incorporated the with plant vector in the transformation. Many other approaches have been utilized the development edible vaccines, which can be categorized in following groups: agrobacterial mediated in the gene transfer, ballistic methods, electroporation. Some of the candidates for the edible vaccines under the study of the edible vaccines, the food include the banana, potatoes, and tomatoes, as well as the lettuce, rice, wheat, soybean, and corn. When choosing a plant to be used as the vaccine, it is the important that the hardy palatable plant with the high nutritive and the protein code. The plant has also one that the would be best indigenous of the country in which is to be the users and should be able to the transfer with the relative easy. Examples of the edible vaccines. Some of the edible vaccines vector used. It is used for hepatitis B virus, tobacco plant lettuce vector used. It is used for the hepatitis B. Norvalk viruses, the vaccine, mainly vector used in the tobacco potato, diarrhea and nausea, stomach cramps. And next one is the vaccine, rabies viruses, mainly the vector is in tobacco. It is used for the rabies and transmissible gastroenteritis coronaviruses, mainly used for the vector is in tobacco and used for the maize, rabbit hematologic disease, mainly disease viruses, and potato hemorrhages. HIV viruses is the vaccine, and mainly the vector is in tomato, mainly it is used for the AIDS. Vibrio cholera, mainly vector is in the potato, mainly used for the cholera. Edible vaccine for the women diseases, hepatitis B, diarrhea, malaria, measles. Mainly, hepatitis B virus is the estimated the approximately 4,000 millions people of the throughout the globe, making one of the most common human pathogens. The hepatitis B surface antigen is used in the vaccine against the hepatitis B. Hepatitis B surface antigen was the first recombinant protein that produced the commercially produced the in yeast. This caused the production. However, the recombination method was the serious concern among the entrepreneurs from the developing countries. These viruses cannot be cultured. Fortunately, with the advanced food vaccine technology, Ornithetins group of the successfully developing the tobacco plants producing the vaccine known as the hepatitis B, with the hope of the developing less expensive the production of the stem system. Tobacco plants are the genetically transferred with the gene encoding the hepatitis B surface antigen linked with the constructive 35S promoter. The maximum level of the HbAg is the transgenic leaf represent the 0.01 of the soluble leaf protein. The expressed HbS-Ag assembled to the virus like the particles resemble the yeast and the serum derived. Hepatitis B is the surface antigen was the able to trigger immunoresponse in mice. The anti-hepatitis B response to the tobacco derived the HBS is the qualitatively similar to the after immunizing mice in the yeast derived commercially producing recombinant HSBAC. Diarrhea, the enterotoxic Escherichia coli, Vibrio cholera causes acute in the water diarrhea by colonizing the small intestine and the producing one of the more enterotoxin, including heat liable enterotoxin. Penetrating structure of the enterotoxin from the E. coli and choleratoxin CT shares the common features. The LTA E. coli has the 125 kilo Dalton, a subunit of the pentamerous of B subunit 11.6 kilodalton LTB. Thus, the plants expressed the protein show the partial penetration in order to the test immunogenic raw potato containing the vaccines where the feed the experimental mice and obtain both the humoral and the musical immune response. Malaria. Malaria is one of the most significant causes of human mortality and the mortality of the worldwide with the 300 to 500 million the cost infected annually resulting 1.5 to 2.5 million deaths. Three antigens are the currently being investigated the development of the plant-based malaria vaccines. Mirojoite surface protein MSP, 4 and MSP 5 from the Plasmodium falciparum and MSP 45 from the Plasmodium OLA. Measles. Measles are the highly contagious the viral disease caused by the paramyxoviruses spread by the air and the including symptoms such as the high fever, skin rash, and the spots. Each year, the almost 1 million of the children died from the measles. In many of the survivors, the weekend of the by pneumonia or encephalitis become the deaf. Recent studies that expression of the pyromoxoviruses surface the protein immovable in tobacco, potato, rice, and the lettuce, the satisfying results. Stopping autoimmunity. In past 15 years, investigators have identified the several cell proteins of the Ilskate automatically. The people produce the type 1 diabetes. The development of the plant-based diabetes vaccines, the potato was attempted. The development of the transgenic potato, tobacco plants, when fed the non-obsessive diabetic mice, showed the increase the levels of the IgG in the antibody associated with the cytokinase. The surplus harmful immune response. Feeding of the vaccines to the most mouse strain the because the diabetic helped the suppress of the autoimmune attack and the prevent the delay of the blood sugar. Transgenic tobacco and potato plants were to produce the express of the capsule protein, Norwalk viruses. The pathogenic viruses cause the epidemic acute gastrointestinal movements. Transgenic tobacco potato plants were to create the capsule in the nerve. Okay, these are the plant tissue culture and genetic engineering. The total syllabus is over. According to our annual examination point of view, 80 marks is there. Eight short question. Remaining is the long questions. Okay.